All right, welcome back. In this second part, we are going to look at how we can enhance crop nutritional quality through environmental optimization. We know that both crop growth and quality are determined by the genetic makeup of the plants and the growing environment. There are differences in crop cultivars grown in open field and in CEA facilities. For example, indeterminate tomato varieties are typically used in greenhouse production. Indeterminate simply means the plants continue growing taller and producing flowers and fruits more gradually over a longer period. And those greenhouse tomatoes are typically grown for fresh market consumption. In contrast, open field tomato production uses mostly determinate varieties, which are often referred to as bush tomatoes, and they are typically grown for the processing market. And similarly, greenhouse cucumber varieties generally grow taller with larger leaves, and the fruits are used for fresh consumption. By contrast, field cucumbers are shorter with smaller leaves, and the fruits are generally used for pickling. And the different varieties also means they oftentimes vary in nutritional profiles. Crop quality is a broad term and includes traits such as shape, color, size that influence a market value, other traits such as the smell, the taste, flavor, and texture that affects the sensory value of those food crops. In terms of nutritional and health value, we have desirable ingredients, including minerals, vitamins, dietary fibers, and bioactive phytochemicals. But there are also undesirable ingredients such as chemical residues, heavy metals, and some specific phytocompounds such as nitrate and oxalates due to their potential adverse effects on human health. And greenhouse and indoor produce typically have higher visual appeal and lower chemical residue, which is a great thing for health. In the next few minutes, we are going to look at how the pre-harvest environmental factors can improve the nutritional and health value of vegetables. We've already reviewed the factors that are important for crop yield and quality. So how do those environmental factors affect plant nutritional value? One of the common mechanisms is through directly affecting plant photosynthesis. For example, the amount of light plant receive or the growing temperature would affect plant photosynthetic rate and the accumulation of photosynthesis. And photosynthesis serve as the building blocks for the biosynthesis of secondary metabolites. Another way for those factors to affect nutritional quality is through serving as signals that regulate the biosynthetic pathways of specific phytochemicals. Oftentimes, photoreceptors are involved in this regulation. Some of the factors such as high levels of UVB exposure or drought stress or extreme temperatures like heat or cold can lead to the accumulation of reactive oxygen species. This in turn promotes the biosynthesis of many beneficial bioactive phytochemicals, especially those with antioxidant properties. Some of those factors can also influence the plant's defense mechanism, although oftentimes there's a trade-off between plant growth and yield and the production of secondary metabolites such as phenolic compounds that will help plants deal with the biotic and abiotic stressors. Factors like root zone temperature, pH, water and nutrient supplies can also affect nutrient uptake, and a lot of those bioactive phytocompounds require certain nutrient elements. Specific nutrients can also act as cofactors for enzymes involved in the biosynthesis of key beneficial compounds. I think the recordings are supposed to be around 30 minutes total, and I'm probably going over time already. And we can discuss the mechanisms through which specific factors affect nutritional quality in class. Here, I'm only going to give a couple quick examples of how light affects the nutritional quality of leafy vegetables grown in greenhouses and indoor farms. So let's look at the light environments in the greenhouse first. Compared to outdoor environments, greenhouse glazing materials and the overhead structures can reduce the light intensity by approximately 30 to 50%. And the spectral quality of greenhouse light is also altered, especially there's typically a lack of UV radiation, including the UVA and UVB region. So this reduction in light intensity can reduce plant growth, leading to fewer flowers and fruits and poor quality. Especially during the winter season, oftentimes we find reduced sugar content and reduced accumulation of bioactive phytochemicals such as vitamin C, anthocyanins, and phenolics. Sometimes there might also be an increase in the accumulation of undesirable phytochemicals such as nitrate under low light conditions. And the lack of UV oftentimes lead to reduced accumulation of bioactive phytochemicals, including phenolics and flavonoids. LEDs are currently the most efficient options for horticulture lighting in both greenhouses and indoor farms, and they are available in many different colors. So potentially we can target specific plant responses through applying specific light colors. A lot of research have found that oftentimes the application of narrow spectrum wavebands can be more effective than full spectrum white light at enhancing beneficial chemicals. Here's an example of a study we did with two red lettuce cultivars, red salad bowl and Josai. I know I said those are red lettuce cultivars, even though they look mostly green, because they were grown under relatively low intensity wet light of a DLI of about 10 mole per square meter per day in an indoor production environment. And this is lower than the 17 mole per square meter per day optimal DLI level for most lettuce cultivars. But it is really expensive to provide higher light level with electric lighting. We had another treatment that grew the plants under a higher level of 14.3 mole per square meter per day, still under broad spectrum wet light. The yield did improve, but the red coloration did not improve enough for the crops to be considered high quality. A different treatment. So instead of increasing the intensity of wet light, we supplemented the crops with narrow spectrum blue light for only 7 days during the end of production stage. So the DRI over those last 7 days was also 14.3, the same as the DLI level of the higher intensity wet light, which was applied throughout the production. You could see that the plants had intense red coloration, which is due to the accumulation of anthocyanins, which is a beneficial chemical with antioxidant properties. So this is consistent with some of the previous findings showing targeted narrow spectrum wavebands might be more effective than broad spectrum wet light at enhancing phytochemical production. There are some excellent papers exploring the mechanisms of those plant responses, especially how exposure to different light conditions that could be low or high light intensity or different colors, how that would change the accumulation of beneficial phytochemicals. So the photoreceptors such as UV B photoreceptor UVR8 and the blue light photoreceptor cryptochrome and the red and far-red photoreceptor phytochromes are oftentimes involved, and it does require a good understanding of which spectral region those different photoreceptors are most sensitive to, and also how those different photoreceptors would differentially affect the accumulation of different phytochemicals. On that topic, here's another study conducted by a graduate student in my group, mainly to compare the relative effectiveness of different light spectrum at enhancing the accumulation of not only anthocyanins, but other chemicals like phenolics and vitamin C. Also, with anthocyanins, we can easily assess the change in accumulations through visual evaluation. That's why it's easier to show the changes using photos. Here you can see the red lettuce cultivars grown under a relatively low level of white light did not have enough red coloration to be considered of high quality, but the supplementation with low level UVB really caused intense red coloration in both cultivars. Among supplemental red, blue, violet, and UVA, blue light tended to be most effective at enhancing anthocyanin in both cultivars. Eventually UVB and blue seem to be most effective, but note that UVB was applied at a much lower dosage, so 3 micromole per square meter per second compared to 60 micromole per square meter per second. This is because UVB is of much higher energy and it's impossible to apply UVB at the same light level without severely damaging the plants. It's simply much more potent. We did a follow-up study to further quantify the optimal dosage required for those lattice cultivars with blue and UVB. So in the previous study, blue was applied at a single level of 60 micromole per square meter per second. And here we have 3 levels at 30, 60, and 90. And you can see as we increased the blue light intensity, there was increased accumulation of anthocyanin, especially in the cultivar Zhosai. In with UVB application, seems a lower dosage of UVB at 0.5 micromole per square meter per second was very effective and may be sufficient for good coloration in those cultivars. In addition to anthocyanin, we also looked at total phenolics and vitamin C accumulation. Overall, for total phenolics, UVB application, which are shown in those last three groups of vertical bars, tend to be more effective than blue light, and within both groups, blue and UVB with increasing dosage, it tends to be more effective at enhancing the accumulation of phenolic compounds. With vitamin C, the response was slightly different, but both UVB and blue tended to enhance vitamin C accumulation, most likely due to a stress response. So this will be the last piece of data I'm going to show you today, and I look forward to discussing with you more in class.